My name is Madison, and today I will be talking about the effects of pesticides and stress hormones on amphibian liver function. The reason for this study is due to amphibian decline worldwide. Amphibians' biggest threat is habitat change and effects of pesticides and environmental toxins. We are finding that sublethal doses of pesticides are easily absorbed through the skin of amphibians and can cause negative health effects. In a recent study, exposure to trace levels of pesticide CPF resulted in altered brain shape and elevated glucocorticoid hormones in tadpoles. Glucocorticoids are hormones involved in the metabolism of carbohydrates, proteins, and fats, and the most prominent glucocorticoid in amphibians is corticosterone or CORT. The release of CORT is beneficial and very important in the regulation of the body. However, in increasing amounts for a prolonged period of time, it can be detrimental. Because of this, it is hypothesized that tadpoles exposed to CPF and CORT will have altered liver morphology and altered carbohydrate storage relative to the controlled tadpole group. In this study, 90 lithobae amphibians were treated with ethanol or control, CORT, or CPF. They were in exposure for 7 weeks and were then euthanized. From these tadpoles, dissections of the brain, liver, gut, and pancreas were made. Data was recorded and used to analyze for morphological changes. Staining of the liver was then conducted with H&E and PAS to determine basic morphology of the structure and to identify carbohydrate content. Looking at the results, the CORT-treated tadpoles had both reduced liver and pancreas mass relative to body mass, which can be seen in figures 1 and 2. With the staining of H&E, basic morphological structures like the hepatic portal triad in figure 3 were shown. Staining with H&E also showed differentiation in the pancreas and liver, with the pancreas staining much darker than the liver. Staining of PAS was used to identify glycogen storage in the liver, and this can be seen in figure 5 by bright pink staining. Using a green reagent counterstain in the procedure also stained the nuclei blue in the red blood cells. From this data, it can be concluded that sublethal CORT exposure shows a consistent decrease in liver and pancreas mass in comparison to the controls. Unlike my prediction, CPF had little to no effect on the morphology of tadpoles. The decrease in liver and pancreas mass does, however, suggest that CORT is breaking down glycogen storage in both organs. For future experimentation, I plan to finish the staining livers with H&E and PAS in order to determine whether the differences in liver mass are due to differences in carbohydrate content. Overall, CORT mediates liver growth during development. I'd like to thank the Woodley Lab Department of Biological Sciences and Bayer School of Natural Environmental Sciences for the support. I'd also like to give a huge thank you to Tribeta for funding these features, which has allowed me to continue this experiment. Thank you again for attending this talk and have a great day.